19. The Rise of Life. In 1953, Stanley Miller, a graduate student at the University of Chicago, took two flasks, one containing a little water to represent a primeval ocean, the other holding a mixture of methane, ammonia, and hydrogen sulfide gases to represent the Earth's early atmosphere, connected them with rubber tubes, and introduced some electrical sparks as a stand-in for lightning. After a few days, the water in the flasks had turned green and yellow in a hearty broth of amino acids, fatty acids, sugars, and other organic compounds. "If God didn't do it this way," observed Miller's delighted supervisor, the Nobel laureate Harold Urey, "he missed a good bet." Press reports of the time made it sound as if about all that was needed now was for somebody to give the flasks a good shake and life would crawl out. As time has shown, it wasn't nearly so simple. Despite half a century of further study, we are no nearer to synthesizing life today than we were in 1953, and much further away from thinking we can. Scientists are now pretty certain that the early atmosphere was nothing like as primed for development as Miller and Urey's gaseous stew, but rather was a much less reactive blend of nitrogen and carbon dioxide. Repeating Miller's experiments with these more challenging inputs has so far produced only one fairly primitive amino acid. At all events, creating amino acids is not really the problem. The problem is proteins. Proteins are what you get when you string amino acids together, and we need a lot of them. No one really knows, but there may be as many as 1 million types of protein in the human body, and each one is a little miracle. By all the laws of probability, proteins shouldn't exist. To make a protein, you need to assemble amino acids, which I am obliged by long tradition to refer to here as the building blocks of life, in a particular order, in much the same way that you assemble letters in a particular order to spell a word. The problem is that words in the amino acid alphabet are often exceedingly long. To spell collagen, the name of a common type of protein, you need to arrange 8 letters in the right order. To make collagen, you need to arrange 1,055 amino acids in precisely the right sequence. But, and here's an obvious but crucial point, you don't make it. It makes itself, spontaneously, without direction, and this is where the unlikelihoods come in. The chances of a 1,055 sequence molecule like collagen spontaneously self-assembling are frankly nil. It just isn't going to happen. To grasp what a long shot its existence is, visualize a standard Las Vegas slot machine, but broadened greatly to about 27 meters to be precise, to accommodate 1,055 spinning wheels instead of the usual 3 or 4, and with 20 symbols on each wheel, one for each common amino acid. How long would you have to pull the handle before all 1,055 symbols came up in the right order? Effectively, forever. Even if you reduced the number of spinning wheels to 200, which is actually a more typical number of amino acids for a protein, the odds against all 200 coming up in a prescribed sequence are 1 in 10 to the 260th. That is a 1 followed by 260 zeros. That in itself is a larger number than all the atoms in the universe. Proteins, in short, are complex entities. Hemoglobin is only 146 amino acids long, a runt by protein standards, yet even it offers 10 to the 190th possible amino acid combinations, which is why it took the Cambridge University chemist Max Perutz 23 years, a career more or less, to unravel it. For random events to produce even a single protein would seem a stunning improbability, like a whirlwind spinning through a junkyard and leaving behind a fully assembled jumbo jet, in the colorful simile of the astronomer Fred Hoyle. Yet we are talking about several 100,000 types of protein, perhaps 1 million, each unique, and each, as far as we know, vital to the maintenance of a sound and happy you. And it goes on from there. To be of use, a protein must not only assemble amino acids in the right sequence, it must then engage in a kind of chemical origami and fold itself into a very specific shape. Even having achieved this structural complexity, a protein is no good to you if it can't reproduce itself, and proteins can't. For this, you need DNA. DNA is a whiz at replicating. It can make a copy of itself in seconds, but can do virtually nothing else. So we have a paradoxical situation. Proteins can't exist without DNA, and DNA has no purpose without proteins. Are we to assume then that they arose simultaneously with the purpose of supporting each other? If so, wow! And there is more still. DNA, proteins, and the other components of life couldn't prosper without some sort of membrane to contain them. No atom or molecule has ever achieved life independently. Pluck any atom from your body, and it is no more alive than is a grain of sand. It is only when they come together within the nurturing refuge of a cell that these diverse materials can take part in the amazing dance that we call life. Without the cell, they are nothing more than interesting chemicals. But without the chemicals, the cell has no purpose. As Davies puts it, if everything needs everything else, how did the community of molecules ever arise in the first place? It is rather as if all the ingredients in your kitchen somehow got together and baked themselves into a cake, but a cake that could moreover divide when necessary to produce more cakes. It is little wonder that we call it the miracle of life. It is also little wonder that we have barely begun to understand it. So what accounts for all this wondrous complexity?